Right, so welcome to MIFOBio, and so I will try to tell you a few things about fluorophores now. And I'll start by this nice super resolution image, and by this statement I make that I think you would all agree with. Like whichever nice fluorescence imaging technique you want to use, you need to start with the choice of the right fluorophore and the right labeling strategy. So that's why I'm here to tell you things about them. The first thing is that when you try to draw fluorophore wish list, you have to care about biology, physics, and also perhaps chemistry. So maybe the most important aspect would be biology, and the fact that your fluorophore and labeling strategy should achieve high specificity. So basically you want to see only the thing you're interested in, and you don't want to see non-specific labeling. The second thing is that you don't want your fluorophore to mess up your target in biological function. For example, if you have a fluorophore which is stuck at the entrance of an active site in an enzyme, and your enzyme would not be functional anymore, then that's not good. And if your target gets mislocalized because of your labeling, that's also not good. And this could happen, for example, if you have oligomeric fluorescent proteins that makes a monomeric target like to aggregate, for example. Of course, if you like to work with live specimens, you want your fluorophore to be compatible with this. And with this, I mean essentially being able to enter passively into your cells or specimens. And very importantly, you want to minimize cytotoxic effects and phototoxic effects, which are induced by light. Okay, when it comes to physics now, so we all know we like bright fluorophores that spit out a lot of photons, but maybe even more importantly, we want to have photostable fluorophores so that in total we can get many, many photons out of single fluorophores. We want fluorophores of many colors if we want to do multicolor experiments, or perhaps we want to use tricks like different behaviors of fluorophores having the same color to produce kind of pseudo multicolor images. So that can be very useful. Often we want to be quantitative. So like we want maybe only one fluorophore per target, and we want like a simple behavior that allows for counting. And finally, when it comes to all these advanced fluorescence microscopy techniques, essentially like super resolution by SMLM, we want smart, so-called smart photophysical behaviors, which has to do with like photo switching, blinking, and so on. In terms of chemistry, so if we synthesize fluorophores, we want these fluorophores to be small, chemically stable. Most of the time we want them to be as insensitive to the environment as possible, which is usually difficult. Sometimes, conversely, we want the fluorophores to be specifically sensitive to one environmental parameter. That's basically more like a sensing fluorophore to pH, oxygen, redox, or whatever analyte. So organic fluorophores should be soluble, and they should, for the chemist, be hopefully easy to synthesize, and for the users, cheap to buy. Okay, so this being said, let's go for a little while into basics of photochemistry. And with this, I will introduce you to this famous Jablonski diagram that hopefully you've seen before. So the idea of this diagram is that if you have a fluorescent molecule, a fluorophore, so it sits in the absence of light into a so-called electronic ground state, S0, which is a singlet state, so that's why S. And in this S0 states, it has also some vibrational levels that typically cannot be reached at room temperature, and which can be reached by some kind of spectroscopy like Raman spectroscopy, infrared spectroscopy. Upon absorption of a visible photon of sufficient energy, the system can jump immediately to a so-called excited electronic state, S1 or S2. And from there, very rapidly, there is a combination of like two processes. One is called internal conversion, and the other is called vibrational relaxation, that brings the system to the lowest vibrational level of the first singlet electronic excited state. From there, two things can happen. To return to the ground state, the system can either fluoresce by emitting a photon, so that's radiative relaxation, or it can like wiggle around and come back to the ground state by a so-called non-radiative relaxation. And the balance between these two routes will actually give us the notion of fluorescence quantum yield. Unfortunately, from time to time, the system has a tendency to jump to the so-called triplet state by a phenomenon called intersystem crossing, which involved spin flipping and which is theoretically forbidden. So that's why it's a rare event. And this T1 state, triplet, is really a bad guy. It's a gate to many bad reactions that produces problems in fluorophores, or sometimes that can be used at advantage. So the triplet state is the origin of the phosphorescence phenomenon, which is a kind of redshifted and delayed emission of light. And the triplet state is also the start of further photochemistry, driving the system to either kind of dormant state, a kind of a coma of fluorophores, eventually being able to wake up, and even worse, the death of the chromophore, which is irreversible. So these events can, to a lower extent, but can actually originate from singlet states also. In terms of timescales, it's important to put these events on timescales. So this absorption is instantaneous. Internal conversion, vibrational relaxation, that's picosecond timescale. Fluorescence, that's nanosecond timescale, and that's very important because nanosecond, that's essentially fluorescence lifetime. It's sense a lot of things in the environment because that's not such a short time. However, the triplet state lifetime can be a lot longer. So it can live, let's say, from microseconds to milliseconds or more. So that's 6 orders of magnitude longer. And this nasty state, so has plenty of time to damage the fluorophore itself or the environment around. And when it comes to dark states, they can live a whole range of times up to seconds and even more. So the fluorescence lifetime, it's basically the rate at which the excited S1 state comes back to the ground state. And so it's described by this little formula, and it's essentially nanoseconds. So little definition about fluorescence quantum yield. So fluorescence quantum yield, that's essentially the probability that a fluorophore will return to its ground state after photon absorption by photon emission. So it's expressed by this little formula here. So it's basically the rate of radiative deactivation divided by all the other rates, which typically can be approximated by the sum of the radiative and non-radiative rates. And for common fluorophores, fluorescence quantum yield is, say, between 0.1 and 1. So close to 1 can really be achieved in real life. We can extend this notion of quantum yield to everything that can happen to a fluorophore. So we call this like 5 phototransformation, where phototransformation can mean like triplet state formation, photo bleaching, switching, whatever. And so we have this notion that quantum yield for a phototransformation is just the probability that this phototransformation occurs after photon absorption. And as few examples, so intersystem crossing, so triplet state production, typically would correspond to a yield of 10 to the minus 3, whereas bleaching would maybe correspond to 10 to the minus 5 or so. So you would need, for example, on average, 100,000 photons before your fluorophore would die. Okay. The notion of brightness, I feel it's interesting to show this graph where you see the brightness of different fluorescent proteins as a function of their color. And what you can see is that, so the brightness, which is the product of fluorescence quantum yield with the extinction coefficient, so propensity to absorb light for epsilon, propensity to emit light from the yield, you can see that we clearly reach a maximum in the green yellow region of the spectrum. And there is a nice rule of thumb to explain this, which is that if you go to the red, basically your fluorophores, they get longer, so longer conjugated systems. So they're bigger in space, so they absorb quite nice. They have a high extinction coefficient, but they are very complex in terms of the degree of freedom they have. So they have a tendency to relax a lot more easily by non-radiative deactivation, meaning that they have a fairly low fluorescence quantum yield. On the other side, when you think about blue fluorophore, they have a small size, and therefore they actually don't absorb very much light, although they can have very high quantum yield. So the best compromise is really achieved in the middle. So another important aspect, which you'll certainly hear about during the school, is photon budget. So this photon budget is essentially the total number of photon that a fluorophore can emit before it undergoes irreversible photobleaching. So here we have kind of an exemplary single molecule trace of a fluorophore, and you can see it emits bursts of fluorescence separated by long dark times, and the total number of fluorescence which are emitted before final bleaching would be the photon budget. And that's super important to define the goodness of a fluorophore in SMLM, for example. So I'll show you a little bit what can happen during photo bleaching and the relation of this with oxygen, which is a very important aspect when people try to photostabilize fluorophores. So I come back to this Jablonski diagram and this notion of the triplet state, which is a gate for photochemistry. And one particular thing that can happen, and does happen very often, is the fact that, so T1 is a triplet state, and it turns out that oxygen, O2, in its ground state, is also in a triplet configuration. So these two, they like each other very much. And so if this fluorophore enters the triplet state, what can happen if O2 comes in is that, boom, they react together and they will produce reactive oxygen species. these reactive oxygen species, like singlet oxygen, for example, can either directly attack the fluorophore itself and it kills it, that's photobleaching, or it can maybe send this reactive oxygen species around in the environment and it damages the environment. And that's also very problematic. So fluorophores are nasty things, especially in the presence of oxygen. So another very important aspect that I touched already a little bit is that due to the long lifetime of fluorophores, nanoseconds, I don't know why there is this line coming, bizarre, fluorescence has the capacity to sense the environment. So there is a long list of experiments that have been designed over the years to take advantage of this sensitivity to the environment. And we can list solvent relaxation effect, solvatochromism, by which a fluorophore which change color as a function of the polarity of the solvent around, for example. Anisotropy effect, where you can, for example, probe the size of a target molecule according to the tumbling time. Like the bigger it is, the more time it takes to tumble and to depolarize light. Quenching effects also, so the fact that you have maybe encounter between a fluorophore and a quenching molecule, and this can reduce fluorescence. And you have all these aspects of energy transfer and fret, for example. So fret is a kind of a channel by which energy can be transferred to an acceptor molecule on a kind of time scale which is equivalent to the lifetime of the donor molecule, so that these two actually compete with each other, and so fret can be used in this way to measure distances at the nanoscale. Okay, so I will now go a little bit into the different families of fluorophores that you will see hopefully during MIFOBio a lot. And I'll start with fluorescent proteins. I'm showing actually this nice image of corals, because a lot of fluorescent proteins were found in corals and anemones, notably the red fluorescent proteins and photoconvertible fluorescent proteins. So in addition to the classical GFP that were found in jellyfishes. So this is GFP fluorescence, and so what you can see here is that fluorescent protein is a little protein of about 30 kilodalton, where a fluorophore, which is actually produced by a maturation reaction from 3 amino acids that are part of the GFP gene. So this fluorophore is embedded into kind of a Coca-Cola-like can, which is specific of typical of fluorescent proteins. So if we zoom in a little bit into the GFP scaffold, we see this fluorophore here, which is made so of 3 amino acids in GFP, so tyrosine, serine, and glycine, and they form, after this maturation reaction, a highly conjugated system that is able to absorb light. If you form this chromophore synthetically and put it in a glass of water, it would absorb but not fluoresce at all. So in GFP, there is a high fluorescence because this chromophore is actually held quite rigidly by a number of interactions with the neighboring environment, electrostatic interactions, H-bond interactions, van der Waals, and so on. We have two main family of fluorescent proteins, just said this before. So the hydrozoan FPs issued from jellyfishes and the anthozoan FPs found in corals, anemones. And they basically look alike, except those ones are a little bit more elliptical. These ones are really round. And so the most distinctive thing is that those are mostly red, those are mostly green. And those FPs are, I would say, a lot more complicated to handle, typically because they are originally very strong tetramers. So it's the difficulty to monomerize them. So You probably know that GFP discovery was also awarded by the Nobel Prize to these 3 guys in 2008, and we have a whole palette of fluorescent proteins. You can do your market, so that's great. So a lot is done with fluorescent proteins. What I want to stress is that fluorescent proteins are proteins, and since they are proteins, they are behaving as proteins. That means they are super dynamic. Although FPs are presented usually as a kind of rigid structure, there is actually motion in there, and it's actually behind the highly complex photophysics that many fluorescent proteins exhibit, that there is all this motion. So I will quickly go through the different kinds of phototransformation that fluorescent proteins may show, but this is also valid for many fluorescent dyes. So it's a kind of a recapitulation. So you get photobleaching, and this is now you know what this is. So the death of the chromophore. Photoblinking, that's very often due to these dark states that come up, and these dark states that can be very long are at the basis of like storm microscopy with fluorescent dyes. And photoactivation now, that's also very useful. You start with a non-fluorescent state, so if you illuminate with light, nothing happens. And with UV activation, you turn the protein irreversibly into an emitting state, being green or maybe red. Photoconversion is the idea that you start with a certain color of fluorescence, typically green, and with UV light, you turn irreversibly the system into a red system that is able to emit red light. So this typically involves light-induced backbone breakage of certain fluorescent proteins that results in extension of the conjugated system of the chromophore. And finally, photo switching, which has also a lot of application, such as, for example, resolved microscopy, which is kind of a similar concept as STED, but employing a lot lower light levels. In this case, illumination at some point will turn the fluorescence on, and this is often involving isomerization of the chromophore. But this is a reversible process now, so with UV light, you can turn the system back on. And this can go on and on for multiple cycles until photobleaching. So a summary for fluorescent proteins follows. So you have certain advantages and certain drawbacks. So I want to draw your attention on these. So the main advantage is listed here. Compatibility with live cells, because they are directly expressed, or by transfection they can be delivered easily in the cell. The labeling specificity is, let's say, near perfect, because of the idea of using fusion genes. There is essentially no non-specific labeling unless you get weird, like protease breakage, but usually it's very high specificity. Stoichiometric labeling, so you get one target, one fluorophore, and that's super nice when you want to do quantitative measurements. And finally, a lot of things have been done with fluorescent proteins to engineer them as sensors. So they can sense a lot of things, like pH, these reactive oxygen species, redox state of the cell, calcium, many others. So they are super useful for sensing. But unfortunately, they have a lot of drawbacks. They are not very bright. They are typically not super photostable. They may not always preserve biological function, because after all, they are not that small, 30 kilodalton. If they are mispositioned, this can pose a problem concerning biological function. And a very critical aspect is the slow maturation. So this maturation of the chromophore from 3 amino acids can take not only minutes, but sometimes hours and maybe even many hours. And this is potentially a serious problem when you do imaging. In some cell compartments, fluorescent proteins may have trouble to fold properly. So you may end up with non-fluorescent fluorescent proteins. You have to care about how you would fuse the fluorescent proteins with a certain target partner. You have to care about oligomerization of fluorescent proteins. So they always tend at some point to make dimers, or when it comes to red proteins, to make dimers or tetramers. So this is a key aspect. And finally, codon optimization is important. There is one thing, last thing about fluorescent proteins. Maybe we hear less and less about them. We can wonder whether it's the end of them, because there are so much progresses on the side of synthetic fluorophores. However, in the last few years, we have seen the discovery of super stable fluorescent proteins, not only like green, like with Stegold and M-Stegold, but also yellow and recently red. And these super stable and bright fluorescent proteins bring fluorescent proteins almost to the level of organic dyes. So maybe it's not the end for fluorescent proteins. Rapidly, I go about synthetic dyes. So there is a huge plethora of organic dyes that are available. So I won't go into the details of these. There are many chemical functions that can be used to produce fluorescent molecules. So you see here a table that has been produced by Luc Levis. You will hear a lot more tonight about this kind of fluorophores. But essentially, what people are interested nowadays is long wavelengths of emission, so red, far red, and high brightness, of course. And so we are interested in this part of the table. And these fluorophores mostly are made of rhodamines and cyanines, I would say. So I'll just say a few words about these two families, which are also the families that are mostly used for super resolution. So we have these rhodamines, like hetero-tricyclic moieties, and we have the cyanines, which are based on polyethylene groups that define a certain color depending on the number of conjugated polymethynes. And so, yes, so there is a huge work done on development of organic fluorophores, and we can cite rapidly. So the engineering of many, many different colors, and a lot go in the red to far red color. Dyes that are fluorogenic, so they only become fluorescent when they bind and at the proper place for labeling. Otherwise, they don't produce any background fluorescence. Dyes that are compatible with living cells that are able to cross membranes. Dyes that spontaneously blink, so they don't necessarily need like a special buffer, like a storm buffer. And this is very nice to perform like SMLM, possibly in living cells. Dyes that are photoactivable or photoconvertible, photoswitchable, exactly that as I presented for fluorescent proteins. And finally, dyes that are photostable, that can actually self-heal in the sense that they can be protected by some attached chemical groups that, for example, eat up the rows that are produced through reaction with the triplet state, and so on. So a lot is happening, and this is quite amazing. So these dyes, of course, they need to be addressed to their target. And here you have also a panoply of techniques. So we've seen fluorescent proteins, which is based on gene fusion. But there is the whole field of immunolabeling that is used for dyes, where you can use like normal like antibodies, primary, secondary antibodies. So that's kind of very widespread, but it's also not that good because this is really huge and the fluorophore ends up being very far from the targets. So it's certainly not the best idea for super resolution. So people have turned to Llama and Camelidae single chain antibodies, and they come up with nanobodies, which are much smaller. And some people also have developed very nice like RNA-based antibodies, we could say, called aptamers or affimers. So a lot of different options. Then what is also very used nowadays are the concept of self-labeling tags or semi-fusion techniques, where you would fuse a target to a tag, a protein tag, that does not fluoresce by itself. But upon addition of a synthetic fluorophore, which is very bright and photostable, you would get binding, either covalent or non-covalent, and this would form a highly performant fluorescent system. And finally, you get many possibilities with click chemistry and non-natural amino acids that can actually be designed to click to an external fluorophore that is brought to the system. This is very nice because then the systems now become very compact. And also, you get this natural labeling. For example, phalloidin, for example, which is naturally binding to actin and to which you can attach a chromophore. So a summary for synthetic dye. Advantages, they are very versatile because they are basically constructed on demand also, almost to our chemistry friends. And so typically they propose, they give high photon budget and very good photo stability. However, most of the dyes still today, they're not necessarily fully compatible with live cells in the sense that they not necessarily cross membrane so easily. So they're mostly used in fixed conditions, followed by permeabilization. And I think a remaining serious issue is the non-specific binding that can occur. And we also need to keep in mind that synthetic fluorophores, they are pretty bad guys in generating garbage, in generating reactive oxygen species. A final slide on the third family. So I really don't go into details, but what you can find on the market, and they have very nice applications, are quantum dots or nanoparticles in general. So quantum dots are essentially like semiconductor nanostructures made of like a core, a shell, and some of amphiphilic surface for biocompatibility. And you have a lot of different nanoparticles. Some of them you can actually take them as a potato bag with a lot of dyes, similar dyes that would kind of talk to each other and build up what we can call a super dye, which has a lot of potential to emit a huge amount of light. So these nanoparticles, they can be engineered in many colors, they can be super stable, and the drawback essentially is their size. They are typically a lot bigger than any other fluorescent proteins or synthetic dye, even attached to their labels. So that's it. I hope I convinced you about the importance of fluorophores, and at the end of the day, very often, I think this is what happens. Like, if you're not really fully happy with the results and you start questioning if your fluorescent marker and your labeling strategy are just the right ones. So you better think twice before engaging into a certain labeling strategy. For those of you that who would be interested in more details about fluorophores in general, there is this school that we organize in March, next March, in Chamonix in France. So if you want to do skiing or mountaineering, hiking, almost as good eating as here, you can subscribe. Deadline is October 30. And with this, I thank you very much for your attention. Sorry.
Thank you for the nice presentation. I just wanted to ask about the development in the direction of bioluminescence. Because,
in the direction of what, sorry?
Bioluminescence. Because, for example, bioluminescent proteins can be useful for tissue imaging, because in this way you avoid autofluorescence.
Yes.
Is there like development in synthesizing bioluminescent dyes, or I mean, also bioluminescence is known for calcium detection, but for more than that, I don't know. So I was wondering.
So in the nutshell, my answer is I don't know. I know that this can be super useful for some experiments, like these luciferase kind of proteins. So they emit at a very low rate, but you don't need excitation, so you can collect light forever without any background. So that's really great. I'm not really aware of major developments, but I must say I don't know. So I'm happy to talk about this with you later. Yeah.